In the last lesson, we learned that there's a certain context dependence to synaptic transmission. The effect of an action potential and its coupling to neurotransmitter release depends heavily upon the recent activity of that axon. Now, in this lesson, we're going to learn that there's another form of context dependence to neurotransmission, that of the neurotransmitter content of the extracellular space. It turns out that the brain is made of about 20% or so of extracellular space, and that then means that neurotransmitters released from one bouton can diffuse relatively long distances and impinge upon membranes that are relatively far from that synapse. Also, interestingly, there are receptors that are present on the presynaptic boutons, so that neurotransmitters diffusing from other boutons can impinge upon these receptors, and that then can affect the release process at this synapse that we're considering. This process of presynaptic modulation then is what we'll be considering in this video. The receptors that are most relevant in terms of regulating presynaptic neurotransmitter release are the so-called metabotropic receptors. These are transmembrane proteins, so they have alpha helices, just like the ion channels, but rather than forming directly an ion channel, they signal through to other proteins that are located on the intracellular side. And so if this is a piece of the plasma membrane, and this is the outside, and this is the inside, then here in the extracellular space, we have neurotransmitters that are diffusing around. Some of these neurotransmitters might then bind to one of these 7 transmembrane receptors. And so they have 7 parts that cross the plasma membrane. These are the alpha helices. When an agonist binds to that receptor, the protein changes its conformational structure, but rather than being an intrinsic ion channel, it then acts upon an intracellular protein, a so-called GTP binding protein, or a G protein. The G protein, upon receptor activation, binds GTP, then it becomes activated. It splits into 2 parts, an alpha and a beta gamma part. They can then diffuse inside the cell, and in part they might act directly upon ion channels. And in part, these G proteins might act upon other enzymes that are present inside the cell, that then might activate downstream signaling cascades. And so one of the enzymes that G proteins act upon is an enzyme called phospholipase C that splits into 2 parts, one part that releases inositol trisphosphate that then goes on to release calcium from intracellular calcium stores, and another part that releases diacylglycerol that then activates protein kinase C. Or we might activate the enzyme adenylate cyclase, and that might increase the level of cyclic AMP. And there are many, many other signaling pathways giving rise to different second messenger systems that affect both the structure of nerve cells and can also even affect the gene expression patterns of these cells. And so metabotropic receptors have very diverse functions inside neurons, and they can therefore be extremely useful. They do, however, operate on a slower timescale than our ligand-gated ion channels that we've been thinking about in terms of synaptic transmission up to now, where we'd have a presynaptic terminal releasing neurotransmitter acting directly on ligand-gated ion channels to create postsynaptic potentials. Now we're thinking about the diffusion of messengers that in itself is occurring on a longer length scale, but also on a longer time scale because diffusion takes some time, and there's then multiple steps in the signaling process. First, we need to activate a receptor that then activates a G protein. The G protein needs to diffuse, and then it might activate secondary messenger signaling cascades. And so the whole process downstream of metabotropic receptors probably requires at least some tens of milliseconds. Probably we're thinking more than 100 milliseconds in most cases, and the effects then can also be longer lasting. They can last for many seconds, and even in some cases, hours. So The metabotropic receptor signaling pathways are extremely useful, very diverse. They couple to many different signaling pathways, and they operate on longer length scales because the diffusion of messengers to these receptors, and also on longer time scales because of the greater complexity of the intracellular signaling machinery that's being activated. There are, of course, a large number of targets that we've already begun to think about in terms of the coupling of the action potential to neurotransmitter release. So if we imagine this metabotropic signaling pathway from the 7-transmembrane receptor activating a G protein, activating other enzymatic cascades, one place where it could interact with is with the action potential waveform itself. We can imagine a fast sodium spike, and the G protein might inactivate potassium conductances, thus giving rise to a broader action potential and more calcium influx, and thus enhancing neurotransmitter release. Or the G protein might directly act on the calcium channel, and thus alter the amount of calcium that enters in the presynaptic terminal. Or it might affect the release machinery or other parts that influence how much neurotransmitter is being taken up, or the recycling and docking and replenishment of the readily releasable pool. So we've already identified many potential targets for modulating synaptic transmission through such metabotropic pathways, and of course it's interesting to see which of these actually occur in a major way in the brain. And one major form of presynaptic modulation is called presynaptic inhibition, and this occurs at almost all synapses in the brain. Near to the release site, but not at the actual active zone, there are typically 7-transmembrane receptors that couple to G proteins. Upon activation by an agonist of the receptor, the G protein splits, the beta-gamma subunit can go and block the entry of calcium through voltage-gated calcium channels, so that's a direct interaction of the G protein with voltage-gated calcium channels to inhibit calcium influx. And the other place where presynaptic inhibition has been noted in a major way is through directly impinging upon the release machinery. And so it inhibits the release of synaptic vesicles in response to calcium. So there are at least two independent ways of regulating neurotransmitter release through presynaptic inhibition. One is reduction of calcium entry, and the other is reduction in release rates in response to calcium rises. So if we now think about how presynaptic inhibition might work in the brain, we can imagine a situation where the neuromodulator, the axon, is turned off, and that we have a presynaptic action potential in a red cell. It propagates down its axon, induces a calcium rise in the presynaptic terminal, and that then, in turn, induces release of neurotransmitter onto the blue cell, driving a postsynaptic potential. Now we turn the neuromodulator on, so there's action potential firing in this neuromodulatory axon, and that axon, the bouton that releases the neuromodulator, is typically not immediately next to the presynaptic terminal, but it might be spaced some 5 micrometers away. The neurotransmitter is then released, diffuses in the extracellular space, so we've got about 20% or so extracellular space, can then bind onto the receptors here at the presynaptic terminal, and then cause a decrease in the amount of neurotransmitter release in response to the action potential. So here's the action potential, here's the postsynaptic potential, and that's clearly smaller than when there was no neuromodulator present. And so this is then presynaptic inhibition. The neuromodulator has caused a decrease in the amount of neurotransmitter released in response to the action potential. So this is a simple view of presynaptic inhibition. The neuromodulator comes on, and that simply decreases the amount of neurotransmitter that's released. However, the situation is not quite so simple, because we also have to think about short-term synaptic plasticity that we discussed in the last lesson. And one common form of short-term synaptic plasticity is depression, where a second action potential induces a smaller postsynaptic potential because of a depletion in the amount of readily releasable pool in the presynaptic terminal. Now, in the presence of a neuromodulator, we've decreased the amount of neurotransmitter released. We decreased the release probability. And so this action potential that here caused a major depletion of the presynaptic terminal now fails to do so. There's only been a small amount release. The neuromodulator decreased the release probability. That then means, in response to the second action potential, that we can get a facilitation. We can get an increase in the amount of neurotransmitter released. The calcium is probably peaking at a higher level, and the first action potential didn't deplete the synaptic vesicles. So they're still all present, or at least many of them are still present. And that then allows the second action potential now to become facilitating rather than depressing. And so there's an interesting interaction between presynaptic inhibition and presynaptic neuromodulation in general and short-term plasticity, where presynaptic inhibition causes a change from depression to facilitating kinetics in general at the presynaptic terminal. There are, of course, very many neurotransmitters that act as presynaptic neuromodulators. Glutamate and GABA are two of the most important ones. These are also the main synaptic neurotransmitters, so they release glutamate directly onto ionotropic glutamate receptors, and GABAergic synapses release GABA directly onto GABA ionotropic receptors. And that's a direct, local, point-to-point neurotransmission that occurs on that 100 nanometer length scale. However, the glutamate and the GABA released at these synapses also diffuse out into the extrasynaptic space. And there they can diffuse for long distances. And indeed, in general, it's thought that the extracellular space has relatively high concentrations of glutamate and GABA that presumably are escaping from their synaptic release sites. And certainly, presynaptic receptors, metabotropic glutamate receptors, metabotropic GABA receptors are equally important in terms of modulating brain function as many of the other metabotropic receptors. And they form an important complement to the ionotropic glutamate and GABA transmitters that are working on the millisecond timescale, whereas the metabotropic receptors are working on the hundreds of milliseconds or second timescale. So glutamate and GABA, in addition to the fast ionotropic action driving postsynaptic potentials, also drive much slower neuromodulatory events, including presynaptic inhibition. Similarly, there are axons for acetylcholine, dopamine, serotonin, and many other neurotransmitters that travel long distances and can traverse long and large parts of the brain. So a single axon from a cholinergic neuron can innervate a large expanse of, for example, neocortical territory. And there, the boutons diffusely release neurotransmitter into the extracellular space. Very often, for the neuromodulatory neurotransmitters, there is no postsynaptic specialization, and the neurotransmitter is simply released into the extracellular space where it diffuses and adds up to a volume transmission of neurotransmitter. And so acetylcholine, dopamine, serotonin, and these other neurotransmitters that you see listed here are all typically working in a volume-like manner. So this means that they don't have the point-to-point specificity of the direct synaptic transmission, but rather they act on a longer length scale and also on a longer time scale to affect presynaptic release and other components of the synaptic transmission process. So the synaptic transmission, the output of an axon, is modulated in a number of important ways. We learned previously about the temporal modulation in terms of the action potential firing pattern of that neuron that can make a big difference in terms of facilitation or depression of neurotransmitter release. Here we now see that the neurotransmitter concentrations in the extracellular space can also affect neurotransmitter release in an important way. And so during different behavioral conditions, acetylcholine, serotonin, dopamine levels will increase or fall in different parts of the brain depending upon the behavioral context, and depending on where the receptors for these neuromodulators are expressed, this will have different and cell type specific effects upon the downstream brain structures and the synaptic transmission at different synapses between different specific cells in the brain. So we've considered the situation where presynaptic metabotropic receptors on the bouton will affect the coupling of the action potential to the neurotransmitter release process. And a prominent feature here is presynaptic inhibition. Now, these metabotropic receptors are not just present on presynaptic membranes, but are also present postsynaptically. And so here, if we think about a postsynaptic specialization and a dendrite that'll hook up to the soma of a cell, there will also be receptors, 7 transmembrane receptors, decorating the postsynaptic membrane. And so we can imagine having a 7 transmembrane receptor here coupling to a G protein, and that then might affect various cellular processes here in the postsynaptic neuron. In part, it might affect similar things postsynaptically as presynaptically. In fact, we also have voltage-gated calcium channels here on the postsynaptic membrane, and 7 transmembrane receptors via G proteins have been shown also to block voltage-gated calcium channels on the postsynaptic dendrites. And so that's one similarity about the signaling processes in the presynaptic and the postsynaptic specializations. Also on postsynaptic membranes, there are other interesting potassium channels that are strongly regulated by 7 transmembrane receptors. So again, via G proteins, they can affect potassium channels. In particular, there's a family of potassium channels called the G protein inward rectifying potassium channels, or the KIR channels, that are strongly affected by G protein coupling here, and they're activated by receptor activation. And this then can give rise to so-called slow postsynaptic potentials, where we can imagine, say, from a GABAergic release of neurotransmitter that can then cause an inhibitory postsynaptic potential that's fast via the standard ionotropic receptors. This is a spike in the presynaptic cell, and it can cause a slow postsynaptic potential through activating potassium channels via this G protein signaling pathway. And this might be something that takes place on the half-second timescale, so much slower than the 10 to 20 millisecond postsynaptic potential via the ionotropic receptors. So there are both presynaptic and postsynaptic effects of neuromodulation, and both of them contribute equally importantly to the neuromodulation of brain function. One of the exciting features about neuromodulation is its clinical relevance. The location of where receptors are expressed to the different neurotransmitters turns out to be highly specific. So some cells express one type of dopamine receptor, others will express another type of dopamine receptor. And similarly for serotonin receptors, adrenergic receptors, oxytocin receptors, you name it, the 7 transmembrane receptors are expressed in highly specific manners in terms of which cells express those receptor subtypes, and also where in the nerve cell those receptors are located. So some will be located on presynaptic specializations, others on postsynaptic specializations, and some will only be located on a presynaptic specialization that contacts a specific postsynaptic cell type. And so where the neuromodulators gain their specificity from is really where the receptors are expressed. The neurotransmitter itself is, of course, also released with some temporal and spatial specificity, but that's much less so than the standard fast synaptic transmitters we've been thinking of. And that means that they're relatively amenable to neuropharmacology, which, of course, we can influence the receptors and the neuromodulatory transmitters on long time scales and without much spatial specificity. And there's some match between what we can do pharmacologically and what is physiologically involved in the neuromodulatory process. And here I'd just like to highlight 2 types of drugs that are in common usage for different brain disorders. One is the fluoxetine sertraline type of drug that works against depression, and the way that they work is as a selective serotonin reuptake blocker. And so we have our serotonin nerve terminals, our boutons that release serotonin. Serotonin is also called 5-hydroxytryptamine, 5-HT. And so these serotonergic axons release serotonin into the extracellular space. There's no postsynaptic specialization for these axons. And the amount of neurotransmitter that's released and the distance it can diffuse depends upon how it's degraded and taken up from the extracellular space. And it turns out that there are specific serotonin transporters that suck up the serotonin from the extracellular space, and that's one of the major ways in which the extracellular 5-HT concentration is regulated, is by the so-called serotonin reuptake proteins. And what fluoxetine and sertraline do is that they block that reuptake machinery, and that then allows the 5-HT to diffuse further and reach higher concentrations. And so fluoxetine has its major way of action to increase serotonin in the extracellular space by blocking the reuptake blockers. And it turns out that the fluoxetine has been very successful in countering depression and anxiety. Another drug, risperidone, acts by blocking specific subtypes of dopamine receptors, of serotonin receptors, and adrenergic receptors. And interestingly, it seems to derive some degree of specificity from not binding to muscarinic acetylcholine receptors. So it targets many of these other systems, but seems to have more than 1,000-fold less binding to muscarinic acetylcholine receptors. And so another drug, this risperidone, acts at a whole bunch of different neuromodulatory receptors, and it seems to have an interesting and useful effect as an antipsychotic that can help treat patients with schizophrenia. The neuromodulators bind to 7 transmembrane receptors that activate G proteins, they bind GTP, they diffuse and activate then downstream signaling pathways. And there's really a large variety of signaling pathways that are open to 7 transmembrane receptors. In particular, they act to presynaptically inhibit neurotransmitter release by downregulating calcium influx and by decreasing the efficacy of neurotransmitter release, and that then also impacts synaptic dynamics in the presynaptic terminal. The neuromodulators are also particularly interesting because they act on this long timescale and in a more diffuse way. They're also amenable to pharmacological interventions. And there's a great deal of interest in trying to specify which receptors are responsible for which types of neuromodulation and how that interacts with different brain diseases, which is an area of intense research in neuropharmacology.